Okay, so we have made it to chapter 29, which should be the second chapter in Unit 2. Unit 2 is all about plants, so this is still about plants, and in particular we're talking angiosperms mostly here. So in this chapter we're going to talk about plant structure and resource acquisition, and then transport, and then a little bit on like plant nutrition and soil type, and kind of atypical plants, I guess you could say. You'll see when we get there. So as far as plant structure and resource acquisition goes, plants need to be able to get resources from both above and below ground. So natural selection has really favored plants that tend to be taller, plants that tend to have flat appendages, plants that have multicellular branching roots, and efficient transport. Plants' algal ancestors, they didn't have those structures. They absorbed their water and minerals and carbon dioxide and nutrients from the water that surround them. And then early non-vascular plants had to live in kind of shallow water and then had aerial roots. But we're not saying like all plants are super tall. Obviously, there's different niches that different plants are going to fall into. But generally speaking, if you're a taller plant, you can outcompete shorter plants for light. If you have big flat leaves, you can harvest more sunlight, and if you have branching roots, you can acquire more resources from below ground. Then vascular tissue, like xylem and phloem, has allowed plants to be able to transport water and minerals from their roots to their shoot for xylem, and then phloem transports the photosynthetic product from a sugar source to a sugar sink. And we'll get more into that in this chapter. I know I said last chapter we'd get more into it, but now this is the chapter we're going to talk about it more. So when we think about plants and their body mass, probably not something you think about too much, but if you do think about it, I think oftentimes people or students might think that like the mass of the plant comes from the soil because, that's where their roots are and that's where they get their nutrients from, but most of their, fresh mass is water, so that does typically come from the soil, but if we dried the plant out, the bulk of their dry mass comes from their carbohydrates that they assimilated from the carbon dioxide in the environment. So the bulk of the plant's dry mass or non-water mass is actually from organic molecules they assembled, and they built the carbon backbone from carbon dioxide in the environment. Obviously, that's not all of their dry mass. About 4% of the plant's dry mass comes from inorganic substances that did come from the soil. As far as how a plant is kind of built, they are adapted to acquiring both light and carbon dioxide, and there's kind of going to have to be compromises between enhancing photosynthesis and minimizing water loss. So if you have too big of a leaf or too broad and flat of a leaf, you're going to lose more water from all that surface area. So there's got to be some kind of compromise between that, and different plants are going to do that differently. That's why they look differently. Plants can do some self-pruning where they shed their lower leaves when they respire more than photosynthesize. What does that mean, respire more than photosynthesize? Photosynthesis is obviously, capturing carbon dioxide and creating organic molecules like glucose or something like that. Respire is talking about cellular respiration. What's cellular respiration? What do you need to do cellular respiration? You need oxygen, and you also need a carbohydrate. So we're saying that the plant gets rid of leaves when they do when they spin more carbohydrates than they produce. And the plant's like, you're not making carbs, you're out. Get out of here. That can also be a moisture loss strategy, which we can get into that a little bit more later. But if you have any house plants and they start dropping their leaves, they might not have enough light, and they probably don't have enough water. So watch out for that. Leaf orientation also affects light absorption. In low light conditions, plants tend to have horizontal leaves because those horizontal leaves can capture more sunlight. In higher light conditions, they're going to have more vertical leaves because those vertical leaves are less damaged by sun, and it allows light to reach the lower leaves as well. I always think of like a prairie when I think of vertical leaves, because like grass, right, has vertical leaves. Although I guess it wouldn't have to be a prairie. It could be like your lawn too. But those vertical leaves are going to be in very sunny conditions, where like your lawn typically is pretty sunny if you can get grass to grow there, or a prairie is very sunny as well, or at least the top surface of the grass is obviously under the grass not quite as sunny. So your root growth can actually adjust to local conditions. Roots branch more extensively in areas where there's high nitrate compared to areas where there's lower nitrate. So if there's a lot of resources in a particular area, the roots will branch out more so they can acquire those resources. And competition is actually reduced within the root system of individual plants, so the plant doesn't try and compete with itself. And an example of that is they did a study on buffalo grass cuttings, and these buffalo grass cuttings produce fewer and shorter roots in the presence of cuttings from the same plant as compared to cuttings from other members of the same species that aren't identical. And then roots can also form mutually beneficial relationships with different soil microorganisms, which, if you remember in Unit 1, we did talk about that, and we'll talk about it more in this chapter, I think towards the end of this chapter. So as far as uptake and movement of materials in plants goes, there's short distance transport, there's medium distance transport, or sometimes people call that tissue level transport, and then long distance transport as well. And we're going to kind of remind you of these, but you've probably hopefully remember your different kinds of passive transport from Bio 160. So like passive diffusion or simple diffusion is sometimes what that's called, or facilitated diffusion where you have a carrier protein involved, or osmosis, which is the movement of water across that cell membrane. And then active transport, which requires the cell to spend ATP to do that transport. And then we're gonna talk about all these, but I just think it's useful to put them all in one slide so you can kind of see where we're going. So on our tissue level transport, you've got transmembrane, symplastic, and apoplastic transport, and then long distance transport we've already talked about a little bit before, but this is mass flow or bulk flow in the xylem and phloem. So as far as passive transport goes, passive diffusion is movement of solute, in this case through a phospholipid bilayer, down a gradient. So simple diffusion or passive diffusion is movement of solute from high concentration to low concentration. I always have to pause on that because sometimes I just say words and they come out. I'm like, wait, did you say that right? So you're going from high concentration to low concentration with passive transport. Facilitated diffusion is very similar, except now you have a carrier protein or a transport protein that solute is going to go across. So this could be a channel or a transporter where it's actually going to bind to the solute, and that'll change the shape of the transporter, and then that releases the solute on the other side of the membrane. And then hopefully osmosis is a review as well. Osmosis is a special case of diffusion where you have the movement of water across a selectively permeable membrane that is permeable to water but not solute. So the water moves in response to differences in solute concentrations. And there's actually proteins in the membrane that can facilitate this as well. So there's aquaporons that allow water to move even more quickly. And we'll talk about water potential with plants here. So another short distance transport is active transport. Active transport, you can have different kinds of active transport, but generally speaking, active transport requires cell to spend ATP to do active transport, and active transport is going to move things from low to high concentration, so against their concentration gradients. Co-transport is a type of active transport where a symporter, which is a protein, is going to transport two substances in the same direction across a membrane. So plants use a hydrogen ion gradient to co-transport other solutes. So they actively transport hydrogen ion or protons against their concentration gradient, and then when the hydrogen ion or proton goes back with its concentration gradient, it brings another molecule along for the ride. So in plants, they establish a membrane potential by pumping those protons, and in animals, we'll talk about this more later, but we have a sodium-potassium pump. But it's really the same sort of idea. So let's look at these two pictures here. Which picture is passive transport? So there's a couple things that would kind of give this away. If we look at the concentration gradient, that's one thing to look at. So on this one, you've got high concentration to low concentration. On this one, there's a lot going on. You have one going from low to high, and the other solute is going from high to low concentration. And you also are spending ATP and going from low to high. So which one is passive transport? This first picture is passive transport. There's still a channel protein here, but other than like the energy required to build this channel protein, the cell doesn't have to spend energy to transport the solutes themselves. In this picture, if you've maybe taken anatomy or physiology, they maybe have talked to you about secondary active transport. So that would be what's happening in the symporter. And then in this other protein here, the hydrogen ion ATPase proton pump, or the proton pump, that is primary active transport because the cell is directly spinning the ATP to pump the proton across, and then this other symporter protein runs off of the concentration gradient of those protons. So the energy for the symporter is found in the concentration gradient of the protons. But anyhow, first picture is passive transport, second picture is active transport. So cellular water content and osmosis. It's a little bit more complicated than we told you in 160 about knowing which way water is going to flow and if water can flow into a plant cell, largely because plant cells have a cell wall, which limits the amount of water that can flow into the cell. So osmosis is still going to depend on solute concentration, but it also depends on the turgor pressure, which is the hydrostatic pressure that increases as water enters the plant cells because the cell wall restricts the movement of water into that cell. I think there's a limit to how much water can rush in. In animal cells, there's not a limit. And what happens to the animal cells if too much water rushes into an animal cell? Right? If too much water rushes into the animal cell gets bigger and bigger and bigger, and then eventually it bursts or lyses. But plant cells don't do that. So we can describe plant cells based on their cytosol content and how close their plasma membrane is to their cell wall. So a turgid cell is a cell that has a cytosol full of water and the plasma membrane is pushed up against that cell wall. And so if you remember hypotonic, hypertonic, isotonic from 160, a hypotonic solution would be like pure water. So hypo means below or less, so a hypotonic solution has less solute. So if we put a plant cell in pure water, that would be putting a plant cell in a hypotonic solution. The cell itself would be hypertonic to that. So the water would rush in from the water, in from the pure water into the plant cell. And I mean, a little bit would go up, but the majority of the water would go in, so it would cause the cell to swell. So we could say the cell is turgid. A plasmolyzed cell would be like a cell in a hypertonic solution, so there's more solute outside of the cell than inside of the cell, so the net movement of water is outside of the cell. That would cause the cell membrane to shrink away from the cell wall. To say it shrinks is kind of bad. It's not like the membrane itself gets smaller, but the cell shrivels because it's lost so much water, and so it's lost its turgor pressure. And then a flaccid cell is when we have a plant cell in an isotonic solution. So the net movement of water in and out of the cell is the same. The cell is not pressed up so far against the cell wall, it's kind of bulging, but it's also not shriveled up and damaged either. So a flaccid cell is just a regular cell at rest in plants. So this next part about water potential, this is really a way to keep track of how water moves. So water potential includes the effect of both solute concentration and pressure, and this is really the potential energy of water. So water moves from a region of higher water potential to a region of lower water potential. And pure water has a water potential of 0 megapascals. A megapascal is just a unit of pressure. If you get into physics, I imagine they'll talk to you more about that. And we can talk about water potential with plants a couple different ways. We can talk about cellular water potential or relative water content. So cellular water potential helps us understand how water can move in and out of cells, and then relative water content is the water status of the entire plant or an organ. And this is something you can actually measure. You can go out into the field and cut like a leaf off or a small stem. You can put it into a chamber and you can add gas into that chamber and see how much gas or how much pressure it requires for water to leave the the leaf or the stem or whatever you cut. So it's something that's actually measurable and kind of useful in ecology and interesting. I think it's interesting. So there's math for this. Typically I don't make you guys do too much math because there's not a math prerequisite for this, but I do think math is useful to talk about because I think it can help you understand what's going on if you don't just shut down and go, oh, math, I hate math. Like it's gonna be okay. So we've got our equation here where water potential equals solute potential plus pressure potential, because we said water potential takes into account both solute concentration and the pressure. So our solute or osmotic potential is due to the presence of solutes in water, and adding solutes always lowers water potential, because if you have more solutes, there's less water available to do work, if you think of it that way. So water with solutes has a negative solute potential, and solute potential is either always going to be negative because solutes are added, or 0 because there's no solutes added. It's never positive. Pressure potential is due to physical pressure. Pressure moves water, and pressure potential can be either positive or negative. A turgid cell, remember that's a cell that's very full of water, has a positive pressure potential. And a flaccid cell has a pressure potential that is less than 1 megapascal. And pressure potential of a solution open to air is 0. So if we look at our picture here, we have a beaker of distilled water. So it's distilled, so there's no solutes in it. So our solute potential is 0. It's a beaker that's open to air, so the pressure potential is 0. So the total water potential here is going to be 0. If we look at this other diagram here, we have a plant cell immediately after being put into distilled water. We're assuming this distilled water is in like a beaker, right? But our pressure potential is 0, and the solute potential of the cell is -2 because there are solutes inside of that cell. Pressure potential is 0 because the water's not pushing against the cell wall yet. However, after some time of being in distilled water, the plant has taken up as much water as it can hold. So now the pressure potential is at +2, the solute potential is at -2, so the total water potential is 0 at this point. So we're not going to have a net movement of water into the cell any longer because the cell has filled up with as much water as possible. Here's another example for you. I've got the equation up here just so you can kind of keep track of it. Here we have an initial flaccid cell. Remember, flaccid is just like the normal cell. Its pressure potential is at 0. Solute potential is at -0.7 megapascals. If we add it into a 0.4 molar sucrose solution, the solution's pressure potential is 0. Solute potential is -0.9. So the total water potential of the solution is -0.9. So the water is going to leave the cell because there's more solutes outside of the cell than inside of the cell. And so you can see that net movement of water is outside of the cell. Another example, here again we have our initial flaccid cell with a pressure potential of 0, solute potential of -0.7, so total water potential of -0.7. And we put it into a beaker of pure water with a pressure potential of 0 because it's open to the environment, solute potential of 0 because there's no solute in pure water. So our total water potential of that beaker is 0 megapascals, and we're going from high to low water potential here. So the water goes into the cell and the cell swells up there. So if we have a solution in an open container that is hypertonic to a plant cell, is it going to have high or low water potential compared to the cell, and why? So what does solute do to water potential? Solute potential is always what? Negative. So it's going to have a low low water potential overall because there's solute present. So there's less water available to do work. According to water potential rules, which way will the water move in the system? Hopefully you know from before, if a cell is in a hypertonic solution, water is going to move out of the cell, right? So water is going to move out of the cell in this case. And then I recorded the answers on this because people tend to get really stuck on water potential. Maybe you're not stuck. If not, awesome. If you are stuck, feel free to shoot me an email. I'm happy to kind of go over this with you more. It doesn't have to be, it's terrible, but it doesn't have to be that hard, but we can work through it if we need more help on it. Oh, I forgot I had another thing. So water moves from a region of high water potential to a region of lower water potential. Then next one here, if a plant cell's pressure potential is 0.2 megapascals and its solute potential is negative 0.35 megapascals, what's the resulting water potential? Well, there's an equation up there. Plug the numbers in the equation, right? So water potential equals pressure potential plus solute potential. So 0.2 plus negative 0.35 equals negative 0.15 megapascals. If we put that plant cell in a beaker of sugar water with the solute potential of negative 0.4, which direction will the net movement of water be, or which way will the water go? So pressure potential of a beaker of water. It's open air, so pressure potential is going to be 0. The potential of the sugar water is going to be negative 0.4, so 0 plus negative 0.4 gives you a water potential of negative 0.4 megapascals. And since real water always goes from high to low water potential, the flow of water would be from the inside of the cell to the outside of the cell there. So we made it through short distance transport. Let's do medium distance transport. So for medium distance transport, we've got transmembrane transport, symplastic transport, and apoplastic transport. Transmembrane is basically going across the membrane. So it's export of material from one cell, followed by import of the same substance to the neighboring cell. Symplastic transport is going across the symplast. So this is movement usually by diffusion of a substance from the cytosol of one plant cell to the cytosol of another plant cell via those connections called the plasmodesmata. The symplast is the inner lining of the cell membrane, and it consists of the cytosol of all the living cells in the plant as well as the plasmodesmata. So if you remember cell junctions from 160, plasmodesmata is the connection from one plant cell directly to another plant cell. Apoplastic transport is movement of solutes along the cell walls and the extracellular spaces and the interior of the vessel elements and the tracheids. And the apoplast is everything that is external to the plasma membrane of living cells. So let's check this picture out here, and we'll label it. So as far as which one is easiest to figure out, I personally think this green line here is the easiest one to figure out because we're going from one cell to another cell directly through the cell membranes here with these little carrier proteins, and so that is going to be C, transmembrane transport. This blue line here is going through the plasmodesmata, so that is going to be symplastic transport, and the red line here is going along the cell wall, so that is apoplastic transport. So we made it from short distance transport to tissue level transport, and now we're going to talk about long distance transport. So that would be something like bulk flow or mass flow is something else we could call that. So this is when you have a large volume of liquids moving down a pressure gradient. This happens faster than diffusion and over longer distances than diffusion. This occurs in the xylem and phloem, and realistically the transport functions of xylem and phloem overlap because you can't just move carbohydrates without them being suspended in water in the plant. But generally, when we think about xylem, we think about movement of water. So there's differences in root and shoot cell water pressure, and the leaves lose water, remember, through the stomata. And so basically you're pulling a column of water up from the roots all the way up and out the leaves in the plant. And then in phloem, you have differences in sugar concentration, and sugar is going to go from a sugar source to a sugar sink. And we're going to go over those in more detail coming up. But this picture kind of gives you literally the big picture on what's going on. So you have sun that heats the leaves, and that causes the plant to lose water from its leaves or its body surfaces, and that water is going to I'm pointing on this slide like you can see, but the water is going to go from the leaves to the atmosphere, and then the water is going to be pulled up all the way from the roots to eventually replace that water. So you have a water pressure gradient that powers the transpirational pull of water from the roots all the way to the leaves. Obviously, it depends on atmospheric conditions, or if it's more humid, this doesn't work as well. And obviously, this depends on the soil water content, because if there's not enough water in the soil, the plant can't pull it out of the soil. But generally, this is how it works. So in the xylem transports water and minerals from roots to shoots in the plant. The absorption of water at the roots takes place at the root hairs, and the concentration of minerals is greater in the roots than the soil due to active transport. So this facilitates the movement of water into the roots. There's parenchyma cells that are alive. There's supportive fibers that have thick walls and may either be alive or dead at maturity. And then there's tracheids and vessel elements that are specialized cells for water transport, and they are always dead at functional maturity and empty of the cytosol. So they're kind of like a straw in a way. So the tracheids are long and narrow with slanted ends. You can kind of see that there. They're slanted, and they have lignin in their secondary cell walls, and they have these pits here, which are non-lignified areas that allow the water to flow from one tracheid to another. Then vessel elements are aligned in a pipe-like file known as a vessel. So here's the vessel element. The whole thing is a vessel. They're larger in diameter than tracheids, which means they can do greater capacity, right? They can move more water, right? A small pipe can move less water than a big pipe. Same idea there. And water is going to flow faster through vessels than tracheids. However, vessels are more vulnerable to embolism, which is a gas bubble. So you've maybe heard of embolisms in humans, and it's a pretty bad deal, right? If you have a gas bubble blocking blood flow, like let's say to your lungs, like a pulmonary embolism, or in your brain, those are bad things. And it's also bad for the plants as well if they have an embolism. However, some plants can self-repair if they have an embolism, because they can get enough root pressure to push up this gas bubble, and you can see this with these tiny droplets of water on the leaves, and that's called guttation. So as far as like how does this all work, okay, so we've got water coming in the roots, water's leaving the leaves, but what about in between? Well, water is cohesive because it's a polar molecule and it's got hydrogen bonding, so water molecules stick to each other. They also stick to the lignified walls of the xylem, and so you've got this column of water, and then you have transpiration at the surface of the leaves, so the water column is going to be pulled up. So some water is leaving the leaves, but since the water molecules are sticking together, they're going to be pulled up in this column. So there's tension exerted on water by evaporation at the plant surface, and it pulls this continuous column of water from the soil. So the really interesting thing about this is plants do not spend energy on bulk flow. That's pretty cool. Like we have to pump blood in our heart or in our body with our heart. Plants do not. They're heartless. So the sun's energy indirectly powers transpiration, and so the plants can rely on energy from the sun to pull water from their roots to their shoots and out their leaves. So here's another picture that shows you this whole idea. So there's the stomata in the leaf, and water is getting pulled from the stomata, from the mesophyll, so the middle of the leaf, from the vascular tissue in the plant, from the stem all the way down to the root, and then water gets into the root by way of the root hairs through osmosis there. Here's another picture, because apparently I felt like you were going to struggle with this. If you look at this one from the bottom, so we could start at water going into the root hairs, into the root, up the shoot, right, and then out the leaf through the stomata, through transpiration. So Number 7, explain the appearance of water in this bag based on what you know about plant physiology. This is supposed to be like a survival technique. So like, I guess if you're ever stranded somewhere and don't have any water but happen to have a plastic bag and plants available and like a string to tie the bag off, you can tie it off and get water in the bag, potentially, depending on the environmental conditions. But I guess if you're stuck, you can try it. So there's water in that bag because transpiration from the plant's body surfaces has pulled water out of the plant. So there you go. Bulk flow is not the same as diffusion because bulk flow is driven by differences in pressure potential, not solute potential like diffusion. Bulk flow occurs in hollow dead cells, not across the membrane of living cells, and bulk flow moves the entire solution, not just the water or not just the solutes, and bulk flow occurs much faster than diffusion does, so kind of keep that in mind. When you think about the stomata, you probably think about water loss because that's what we focused on in 160. So it's good that plants lose water from the stomata to some degree because that pulls water from the roots through the shoots and all of that. However, you want to limit water loss because if you have too much water loss, then that's obviously detrimental. So the stomata are the pores in the leaf, and then around the stomata you have these guard cells. So the guard cells actually open and close the stomata. When the guard cells are turgid or full of water, then it's like a full balloon in a way that opens the stomata. When the guard cells are flaccid, that closes the stomata. Some stimuli for opening of the stomata would be light. Why would the stomata open with light? So what's the plant going to do when there's light available? Or what could it do? It could do photosynthesis, right? What does the stomata have to do with photosynthesis? Well, that's how they capture carbon dioxide. You need carbon dioxide and light for photosynthesis. So also stomata open when there's low carbon dioxide levels inside the leaf. And then the stomata can open on a circadian rhythm. So I said stomata open with light, but would a can plant stomata open with light? No, because can plants, remember, open their stomata at night and they do their initial carbon capture in the evening. So there's different triggers for stomata opening, kind of to some degree depending on what exact kind of plant you are. Stomata are going to close when there's drought stress, because if you're losing too much water, you need to close the stomata to try and conserve water. And then they're also going to close in response to a hormone called abscisic acid, which is produced in response to water deficiency as well. So if we look, there's a role of potassium ion in stomatal opening and closing. So potassium ion in these pictures is represented with the red dots there. And so if there's a high concentration of potassium ions inside of the cell, that causes water to follow the potassium, and that causes the cell to become turgid, which opens the stomata, and potassium can actually be moved out of the cell to get water to follow out and close the stomata. So other things plants can do to prevent water loss is drop their leaves. Again, if you have houseplants, this might be something you've seen before. Maybe not. Maybe if you're good at taking care of them, you don't see this, but definitely it's a thing that can happen. So this is a valuable adaptation for desert plants and plants in seasonally cold habitats. It avoids very low leaf water potentials and dangers of xylem embolism, and it reduces the amount of root mass that the plant has to produce in order to obtain water under dry conditions. So here's a desert plant with its leaves on it, and then without the leaves. It's not dead here, it's just dry. So then long distance transport in the phloem is a little bit different than the xylem because the phloem is actually alive at functional maturity, and in phloem cells retain at least some of their cytoplasmic components. So you've got supporting fibers, you've got parenchyma cells, you've got sieve tube elements, and you've got companion cells. Sieve tube elements are going to lose their nucleus and most of their cytoplasm so that bulk flow can occur quickly through them, but they're supported by companion cells that supply their mRNA and their proteins by way of those connections called the plasmodesmata. And then sugar is going to travel from a sugar source to a sugar sink. The sugar source can either be an organ, that stores sugar, or something like a leaf would be a good example of a sugar source because it's producing the sugars by photosynthesis. So here's a nice picture that shows you the sieve tube element and the companion cell. You can see this like tiny little bit of cell membrane here on the sieve tube element, and it's supported by the companion cell there. So the companion cell plays an essential role in conveying sugars to the sieve tube element for long distance transport, and they can enhance solute movement between the apoplast and the symplast. Sucrose is typically what plants are going to use for most long distance transport, and there's two types of phloem loading, either symplastic or apoplastic phloem loading. So with symplastic phloem loading, many woody plants transport sucrose from sugar-producing cells of the leaf mesophyll to companion cells and then to the sieve tube elements by way of their plasmodesmata. What are the plasmodesmata again? Those are those connections from one cell to another where their cell membranes are connected. So this doesn't require ATP because these are just kind of pores from one cell to the next, and because they're directly connected, the sugar doesn't actually have to cross a cell membrane here. So in this picture we've got a sugar producing cell with little sucrose molecules, and it's going from the plasmodesmata or through the plasmodesmata from the sugar producing cell to the companion cell, and then from the companion cell into the sieve tube element of the phloem. So then there's partially apoplastic and partially transmembrane transport. This is going to load sugar into the sieve tube elements or companion cells from intercellular spaces, so spaces between cells. So in this case, ATP has to be used to move the sugar across the plasma membrane into a companion cell or a sieve tube element. So here we have our sugar producing cell, and then here we have some intercellular spaces, so spaces between the cells. So the sugar left the sugar producing cell, it's in this space now, and we have a hydrogen ion sugar co-transporter, and that takes energy because we have to set up the hydrogen ion concentration gradient with ATP here. So that transports sugar into the companion cell, and then from the companion cell into the sieve tube element. So that brings us to the pressure flow hypothesis. This describes the process of loading sucrose into the sieve tube elements at the source. So at the source, where there's a high sugar concentration, the sieve tube elements near the source tissues have comparatively high solute contents, and then water is going to rush in from nearby xylem, which builds turgor pressure, and then at the sink, the sieve tube elements near the sink tissue have lower solute concentration, so water that contains solutes moves from the phloem into the sink tissues, and at the sink tissues the sugar is going to be unloaded through those plasmodesmata, which, what are the plasmodesmata again? Those connections directly from one plant cell to another. Sometimes there's more sinks than can be supported by sources, so the plant is going to self-thin those out by dropping the sugar sinks. So maybe if you're gardening, if you've ever seen a plant like drop flowers or fruits, or even seeds before they're mature, that could be because it just doesn't have enough leaf mass to support that carbohydrate-rich structure there. So we're going to watch a video.
It's summer, and this plant's leaves are making sugars that are being stockpiled in its taproot. A tissue called phloem transports the organic products of photosynthesis throughout the plant in a solution called phloem sap. This process is called translocation. In the summer, which part of the plant is the sugar source? The leaves are the sugar source, producing sugar via photosynthesis. Since the taproot is storing sugars, it is the sugar sink.
Sugar produced by the leaves is pumped into the sieve tube members of the phloem by active transport. This raises the concentration of sugar, causing water to move into the phloem by osmosis. Water pressure at the sugar source increases, pushing water and sugar away from the sugar source and toward the sugar sink, a process called bulk flow. At the sugar sink, root cells remove sugar by active transport and convert the sugar into starch. Again, water follows by osmosis. This lowers the water pressure at the sink. Thus, the phloem transports water and its sugar cargo from the sugar source in the leaves, where there is a buildup of pressure, to the sugar sink in the root, where the pressure is lower.
So here's a picture showing you the same thing as that last video. It's showing you kind of how the xylem and phloem are working together through this process. So here we're at a sugar source, the leaves, where they're actually producing the carbohydrates. In this case, we're going to transport it as sucrose. So we're going from the sugar source to the companion cell into the phloem. Now we have a high concentration of solute in the phloem, so water is going to go from the xylem to there, and then that's going to push that sugar down. The companion cells are going to take that up. The sink cells are going to take that sugar up, and then they're going to create starch, which is too big to go through the cell membrane. And so now we're going to have a lower concentration of solute, so the water is going to hitch a ride on the xylem and back out or up the plant and out through the leaves through transpiration. So you can see how the xylem and phloem are ultimately connected here with bulk flow. So that brings us to kind of another topic entirely. If I was writing the book, I might have 2 chapters on this, but the book didn't. So we've also got macro and micronutrients in this part of the book. So really macro means like large scale, micro is small scale. In this case, we're not talking about the size of the nutrients themselves, but we're talking about how much the plant needs. So more than 50 inorganic chemical elements are found in plants, but not all of them are essential for life. There's 17 essential elements which a plant needs in order to complete its life cycle, and we found out what's essential or not essential by growing plants in hydroponic culture. So you could remove certain nutrients and see if the plant can complete its entire life cycle. And they still may be able to live without certain nutrients, but they maybe can't flower and fruit and produce seeds without all the nutrients. So there's 9 macronutrients: carbon, oxygen, hydrogen, nitrogen, phosphorus, sulfur, potassium, calcium, and magnesium. And there's 8 micronutrients, which include chlorine, iron, manganese, boron, zinc, copper, nickel, and molybdenum. And oftentimes these micronutrients are going to function as cofactors, which are kind of like non-protein helpers that help in enzymatic reactions. I wouldn't worry about memorizing this table, but some of this is pretty obvious, I would say. If we look at the macronutrients in plants, plants need carbon, right? There's carbon in all of their organic molecules. I do want you to know kind of like where they're getting this nutrient from and things like that. So carbon, pretty obvious. All of your organic molecules and the plant's organic molecules need carbon. Plants get their carbon from the atmosphere. Oxygen, again, plants need oxygen because they need to do cellular respiration. Plants need oxygen because it's part of their organic molecules. Same with hydrogen, and kind of so on and so forth there. So don't worry too much about memorizing this table, but I just kind of want to give you a little bit of information about macronutrients in plants. If you take a soils class, you might go more into detail on this, or any kind of ag type class or a plant physiology class. If you take plant physiology, you'll probably get to do some hydroponic experiments and look at plant nutrient deficiencies. As far as deficiencies go, the symptoms of the deficiency depend on whether the mineral is mobile or not in the plant, or the nutrients mobile in the plant or not, and then also what the nutrients functions are. The deficiencies of a mobile nutrient usually is going to affect older organs more than younger organs, so older leaves more than younger leaves, because that nutrient can be taken from the old leaf to build new leaves. And deficiencies of less mobile nutrients usually affect younger organs more than older ones, because again, they can't be taken from older organs and moved to newer ones. The most common deficiencies are nitrogen, potassium, and phosphorus. I colored the text there because they tend to have sort of distinct patterns and distinct colors that you'll see if you run into these sorts of deficiencies. And if you have a certain deficiency, it might not be just because there's not enough of that nutrient in the soil. Like adding more nutrients to the soil wouldn't necessarily fix it. It can sometimes, but it might not. It could also be like if the plant doesn't have enough root surface area available and things like that. So as far as soil management goes, we could get into like a really long thing about soil management, but we're just doing like a little tiny introduction to this. And if you ever have to take a soils class or get to take a soils class, you'll get to go through that in more detail. But according to our textbook, about 30% of the world's farmland has been having reduced productivity because of soil mismanagement. And soil management would include things like fertilization, altering the pH, altering the texture, and then also keeping the soil ecosystem together. So in natural systems, nutrients are recycled through the excretion by animals and the decomposition of kind of all the dead organic material or the humus. And fertilization can be used to replace mineral nutrients that have been lost from the soil. Our book talks about commercial fertilizers and organic fertilizers, although it's not like you can't buy organic fertilizers, you can. But commercial fertilizers would be enriched in nitrogen, phosphorus, and potassium, and excess minerals can leach from the soil and cause harmful algal blooms, which we'll talk more about in the ecology unit. But basically, if you have a high concentration of certain nutrients, particularly phosphorus, that can lead to algae overgrowth, and then those algae die, and then bacteria eat the dead algae, and then use up the oxygen, and then things that require oxygen are not going to be able to live in that body of water. So that would be a bad deal. Organic fertilizers can be composed of things like manure, fish meal, or compost, and they're going to release nutrients more gradually as they decompose. So soil pH is important because it affects cation exchange. What's a cation? It's an ion with a positive charge. So cations are more available in slightly acidic soils because hydrogen ion is going to displace the mineral cations from clay particles. Clay particles have a negative charge, so that negative charge clay particle attracts the cations. But when hydrogen ion comes in and displaces it, those nutrients are more available to plants. So the availability of different minerals varies with the pH of the soil, but at a pH around 8, plants can absorb calcium, but not iron, for example. That was a hard one to say. So here in this picture you can see the ions being displaced by protons or hydrogen ions there, and we've got this big giant clay particle there. So really, some people might say like a good farmer doesn't just grow crops, they grow soil as well, because the soil is an ecosystem. So plants obtain most of their mineral nutrients from the topsoil, and the basic physical properties of soil include the texture and the composition of the soil. When we talk about soil texture, we can talk about soil particles. You've got sand, which is the largest particle, then you've got silt, which is in between, and then clay, which is the smallest particles in soil. So there's a picture over here with sand and silt and clay particles. So topsoil is formed when mineral particles are released from weathered rock, and it mixes with the living organisms in the decaying humus. And the soil solution consists of water and the dissolved minerals in the pores between the soil particles. So soil can be classified by the amount of coarse or fine materials, and we could call soil like sandy or silty or clay or loam soils. And loam soils are the most fertile topsoils and contain equal amounts of sand, silt, and clay. And loamy soils retain water and have air spaces for oxygen diffusion to the roots. And sandy soils have poor water retention, and then clay soils have poor drainage. As far as your topsoil composition goes, the inorganic components of the soil include your cations and anions, and most soil particles are negatively charged, and these negatively charged soil particles are not going to bind with the clay, so they're going to be easily leached from the soil. But cations like potassium or calcium or magnesium can adhere to the negatively charged soil particles. The organic components of the soil include decomposing material like leaves and feces and formerly living organisms, and we call all of that decomposing kind of organic material humus. And humus forms a crumbly soil that helps to retain water but is still porous, and it increases the soil's capacity to exchange cations and serves as a reservoir for mineral nutrients. So we've got another video to watch about minerals from the soil.
Using this cross-section through a root, we can review the way root hairs take up minerals from the soil. First, we enlarge the root hair for better visibility. Roots use ion exchange to detach positive ions from negatively charged soil particles. Root hairs achieve cation exchange by releasing molecules that react in the soil solution to produce H+ ions, which displace the soil ions. Root hairs make carbon dioxide molecules during respiration. Carbon dioxide molecules diffuse into the soil where they combine with water to release hydrogen ions and carbonate ions. The H+ ions bind to soil particles and displace positive mineral ions. Positive ions are called cations, so this process is called cation exchange. Roots also spend ATP to pump hydrogen ions directly from the root to the soil. Soils hold some minerals more tightly than others, and very heavy rains can wash or leach the more loosely held minerals out of the soil. Soil particles tend to be negatively charged and do not bind negative ions such as nitrate. Positive ions stick to the negative soil particles and resist being leached away. Air pollution can cause acid precipitation, in which raindrops carry H+ into the soil. Knowing that roots release H+ into the soil to help the root hairs collect minerals, why wouldn't you expect acid precipitation to make soil more fertile? Acid precipitation duplicates the cation exchange effect of roots. H+ displaces positive mineral ions from negative soil particles. However, the flowing water washes the detached ions away before they can diffuse into the root hair. This makes the soil less fertile.
So make sure you know that acid rain is kind of a double whammy because it washes nutrients away and changes the pH of the soil there. So that brings us to living components of topsoil. So the soil is alive or should be alive. So this includes bacteria and fungi and algae and other protists and insects and earthworms and nematodes and even the roots of the plants themselves. And these organisms help to decompose organic material and help to mix the soil. And then when they die, they become part of the humus as well. Plants and soil microbes often have a mutualistic relationship. Dead plants provide energy needed by soil-dwelling microorganisms, and secretions from living roots can support a wide variety of microbes in this kind of near-root environment. Soil bacteria can benefit plants by exchanging beneficial chemicals with the plant roots or increasing nutrient availability by decomposing dead organic materials and even converting nitrogen. So the soil layer surrounding the plant's roots is called the rhizosphere. Rhiza is root, right? So the area around the root is the rhizosphere. And non-pathogenic bacteria or fungi called endophytes can live between cells within the plant, and free-living rhizobacteria can inhabit that rhizosphere. So rhizobacteria and endophytes depend on sugars and amino acids and organic acids that are secreted by the plant's roots, and they can enhance plant growth by producing growth-stimulating chemicals, producing antibiotics that protect roots from disease, and actually there's a whole potential for antibiotics in general from soil bacteria. We aren't as good at culturing bacteria that live in the soil yet, but we're getting better and better techniques for that all the time. But basically, these bacteria are fighting each other for space and resources, and one way to fight off other bacteria is to kill them with antibiotics. So not too surprising when you kind of think a little bit more about soil bacteria producing antibiotics, but that could also protect the plant from disease-causing bacteria. The bacteria can also absorb toxic metals and use that as part of their metabolism to protect the plant. And it can also make nutrients more available to the roots of the plant. So bacteria are involved in the nitrogen cycle. The nitrogen cycle transforms atmospheric nitrogen gas and nitrogen-containing compounds. Plants can only absorb nitrogen as either nitrate or ammonium, and most usable soil nitrogen comes from the action of soil bacteria. Nitrogen fixation is the conversion of nitrogen from N2 to NH3. And some nitrogen-fixing bacteria are free-living, and others form really intimate association with the plant roots and actually live within the plant roots. So there's different ways that ammonium ion can be produced. So there's ammonifying bacteria that break down organic compounds and release ammonium. There's nitrogen fixing bacteria that convert nitrogen gas into ammonia, NH3, and then ammonia, NH3, combines with hydrogen ion to form ammonium, which is NH4. There's also conversion to nitrate ion that can occur. So nitrifying bacteria can oxidize ammonium, NH4, to nitrite, NO2, and then the nitrite to nitrate, so NO2 to NO3. And there's different bacteria that mediate different steps. And then nitrogen is lost to the atmosphere when the denitrifying bacteria convert nitrate, NO3, back to N2, or nitrogen gas there. So I'm not super worried that you can tell me all of those steps, but we'll get more into the nitrogen cycle when we're in the ecology unit, but I want you to know that there's different bacteria along the way and that only some forms are accessible to the plants themselves. We said that some bacteria form kind of an intimate relationship with plants. This should be a review from Unit 1 when we talked about the rhizobium bacteria. So rhizobium bacteria live in nodules of legumes. A legume would be like a bean plant, but they're not all beans, but that's just a good example of a legume that you maybe have heard of before. So these nodules are infected by these nitrogen-fixing rhizobium bacteria, but to say they're infected doesn't mean it's bad that they're there. And inside the nodule, the rhizobium forms bacteroids, which are going to be inside the vesicles of the root cells themselves. So fungi also can help plants. Mycorrhizae, so myco is fungi, rhizae is root. These are the mutualistic associations between fungi and the roots of a plant. This increases the surface area of the plant because you've got the fungal hyphae there, and they can also stimulate the growth of the roots themselves. Seeds can be inoculated with fungal spores to promote the formation of mycorrhizae. You can actually literally buy this on the internet. I mean, you can buy anything now, right? But you can buy mycorrhizae online if you want. And some invasive exotic plants actually disrupt the native plants' mycorrhizae. They basically can kill the mycorrhizae fungi, which then prevents the native plant from doing as well. So for example, garlic mustard slows growth of other plants because the garlic mustard prevents growth of the mycorrhizal fungi. So here's a couple pictures showing you different kinds of mycorrhizae. You can have ectomycorrhizae or endomycorrhizae. Ecto is outside of, endo is inside of. So the ectomycorrhizae are outside because they're not in the cells, they grow between the cell walls. The endomycorrhizae or vascular mycorrhizae live inside of the cells to some degree there, and they form arbuscules. So some plants have nutritional adaptations that use other organisms in non-mutualistic ways. These include epiphytes, parasitic plants, and carnivorous plants. And these are not typical, they're pretty unusual, but they do occur, so it's worth talking about. So a parasitic plant is a parasite of another plant. It absorbs water and sugars and minerals from another plant. Some species are capable of photosynthesis, but others rely totally on their host plant for sustenance. And some species even parasitize the mycorrhizal hyphae of other plants. So mistletoe here is a parasitic plant, however it is photosynthetic. And then you've got dodder, which is this orangish stuff here. If it's an orange plant, is it doing photosynthesis? No, because what's it lacking? Chloroplast. But why would you make chloroplast if you didn't need to? So dodder is not photosynthetic. It's still a plant though, but it's absorbing nutrients from a host plant. Here's another non-photosynthetic plant. This one happens to be a parasite of mycorrhizae. It's called Indian pipe, and it's actually a pretty interesting plant if you ever see one out in the wild, right? So some people try and collect them and take them home, but it's not going to work for you or anyone. No, it won't. So if you ever see one, just appreciate that you saw it and think wow, that's really cool. But you can't take it and transplant it into your home garden, typically that's going to be illegal, number one. But number two, it won't work and you'll kill it because it is living off of the mycorrhizae of another plant. And so you're not going to be able to get that mycorrhizae in a big enough quantity, right? And get that other plant that's attached to in order to keep this Indian pipe alive. So don't ever dig one of those up and try and transplant it. Just appreciate that you were lucky enough to see it. Epiphytes are other plants in a non-mutualistic relationship. So what they do is they live on other plants. Typically they're going to be gathering their water and minerals from rain rather than tapping into their host plant. So epiphytes don't usually harm the other plant. So they're in a commensalistic relationship. So the epiphyte benefits because it's elevated up, so it gets better light, but it's not harming its host plant. Then carnivorous plants are still photosynthetic, and that seems to be kind of a confusion oftentimes, but they're still photosynthetic, they're still green, they do photosynthesis, but they obtain nitrogen from killing and digesting prey. So I think the most kind of famous carnivorous plant is the Venus flytrap, but there's other carnivorous plants as well. They tend to live in really nitrogen poor soil, so they have these adaptations to get nitrogen from their prey. So let's match the terms to their descriptions. If you want to go ahead and pause this first, feel free. Otherwise, I'll just kind of go right through and match it up here. So number 8 says relationship between roots of plants and fungi that increases surface area of the roots. Roots and fungi would be mycorrhizae. So 8 is E. 9 says absorb nutrients from a host plant, depending on the species, may not do photosynthesis. What's it called when they steal nutrients from another organism? That's going to be C, a parasitic plant. 10, grow in poor soil, usually lacking nitrogen, and trap animals or protozoans. That's going to be B, the carnivorous plant. Grows on the surface of a plant and obtains moisture and nutrients from rain or air. That's going to be an epiphyte, so 11 is A. And 12, nitrogen-fixing bacteria associated with plant roots. That's going to be the rhizobium bacteria, so 12 is D. So we made it to the end of chapter 29, which is really kind of like 2 different chapters in my mind. We talked about transport stuff, and then we talked about plant nutrition and soils. So if you have any questions, as always, feel free to reach out.